everybody stay on board with the innate system so they don't get behind with the onslaught of the adaptive system.
With exams, since there's more immunology, will the exams be more on that opposed to?
Yeah.
Okay.
Will probably be about 10-15% of the patterns, and the rest will be vast amount of difference in information. So remember, this is a very, very important slide. Helps to put everything in perspective with what belongs to what. Okay? So again, if you just look at the boxes over here, the first box is our first line of defense. Let's keep everything from even gaining access to the body. Second line of defense is going to be that internal mechanism of, whoops, the gates broke down, something got in, now how am I going to kill it? That's our second line of defense. The last two boxes together make up the third line of defense. Okay? And that's what we're going to be spending time in today, is talking about the cellular immunity. So, we've got through all of the stuff on fever, and complement, and all that wonderful stuff. So with our adaptive system, remember that the innate is referred to as nonspecific, primarily because it doesn't care what the bad guy is, as long as we know it's a bad guy. It's not self. Remember, that's the whole process and the premise of our immune system, self from not self. Okay? Innate just says, if you're not me, I'm going to kill you. Whereas in the adaptive, it's specific. Okay? And that's the other term for it. It's called the specific. Because it's a one-to-one relationship. For every good guy, there's exactly one bad guy. And that's how it's set up. Now, there's two other characteristics that go along with this system. Besides being specific, this is systemic. Remember, I could do something to my thumb and have an inflammatory process just in my thumb. And one of the goals of the inflammatory process was to isolate it, to prevent it from going global, systemic. Well, the adaptive system is systemic. So even though it still might be fighting a battle here in my thumb, the response is systemic. And then it also has memory. So that once I fight a battle for the first time, it takes a little bit of time. I got to figure out who it is I'm fighting. I got to rev up my battle plans, and then I start the battle. The second time, the third time, the fourth time, it's a much quicker and much bigger response. Oh yeah, we've encountered these people before. I know how to kill them. Boom, we're going to start it up very, very quickly and just eradicate them. Now, the two parts of this are humoral. Now, hopefully you remembered some of the terms we talked about in previous chapters, like the aqueous humor, the vitreous humor. Humor means fluid. So this is something that is part of the liquid part of our blood. It's not a formed element. These are our antibodies. So our B lymphocytes would be contributing to our humoral immunity. And then we have cellular immunity. Cellular immunity is where we have cell-to-cell combat. Again, one good guy for every one bad guy. So our B cells and our antibodies, when we get there, they don't actually kill anything. Well, the B cells can. They can undergo phagocytosis. But for the most part, they're producing antibodies. And the antibodies don't kill. They facilitate other aspects of our immune system of killing. But I do have cellular mediated. So I do have some cells that are able to go directly to a cell and kill it. And again, we'll talk about that today as well. Now, we talked about antigens, and I've used the term many times when we went over histology. And remember that antigen comes from antibody generating, antigen. Okay? Now, for the most part, these are molecules. They tend to be very, very large. When we talk about them here, we're going to talk about them as being something that's exogenous, something from outside of our body. We can have antigens that exist in our body. Okay, and again, we'll talk about some of the pathology that lets us arrive at that particular process. Now, I'm going to talk about antigens when in reality I should be talking about antigenic determinants. Okay? Now, this is one of the reasons I really like the Physio Suite. So, they would say, they describe an antigen as being a person. Okay? And an antigenic determinant would be the nose. In fact, they have a whole little thing where the guy comes by, he's blindfolded, and he goes up to each person and he squeezes their nose as he walks down the line. Because what he's trying to find is he's trying to find somebody that either doesn't have a nose or has a nose that he recognizes. Okay? So, an antigenic determinant looks like this. So if you notice, this is an antigen here, but it's got these things that are called epitopes or antigenic determinants. These are the things that are being recognized as being self or not self. Now, since it's an antigen, more than likely they are going to be not self. Most antigens have multiple different types of antigenic determinants on them. So sometimes we get lazy and we just talk about an antigen. Well, again, an antigen is a molecule that's going to have multiple things that get recognized. So I'm going to put this all in perspective. If I say that, remember there's one good guy for every one bad guy. So this antigen really is composed of 3 bad guys. 1, 2, 3, because they're all different. Therefore, this particular antigen would have 3 good guys, 3 cops after them. One would be after this, one would be after this, and one would be after that. The more antigenic determinants, the more different antigenic determinants that an antigen has, the more or the bigger the response will be. So if you think about, let's talk about a gang. Let's say this is a gang, and these are individual members of the gang, and these are the police. Well, if they're all together, then I'm going to have a bigger response because there's going to be more police coming to this gang because of the different antigenic determinants that are on them. Now, things that we have a tendency to stick in bodies, plastic, metal, when we take care of our patients, so implants, they generally have one antigenic determinant. They have a lot of them, but it's the same one. So they tend to be non-reactive. So it's the ones that have all of the different antigenic determinants are the ones that tend to be more virulent. In other words, they cause a greater amount of disease. Now, remember that we talked about this flagpole, the major histocompatibility complex. And what hangs off of flagpoles? Flags. And those flags, again, so this would be an MHC. That's the flag. And it's either going to say self or not self. And again, now that we're in the adaptive process, there's also going to be those little symbols that I talked about. Which is basically, this is an antigenic determinant. So depending on what's on the flagpole, it's going to designate what ends up happening to this cell. Now when I say a flagpole, again, this is going to be attached to a cell. So there might be an antigenic determinant, which is what this lower part stands for, it's going to cause a response. Again, the innate system, non-specific, just looks for the letters. Adaptive looks for the symbols. And again, the symbols don't really exist there. These are just simply peptides from something that's been broken down. Now, I will tell you that antigens can be what are referred to as complete or incomplete. And this is a thing that tends to be somewhat confusing for students to get on first go around. For something to be an antigen, it has to have what's referred to as immunogenicity. In other words, it causes an immune response. It causes antibodies to be formed. The second thing is called reactivity. The antigen would then react with those particular antibodies that were just formed. That's called a complete antigen. We have things that are referred to as incomplete. I had to go in reverse there for a minute. Incomplete antigens, which are called haptens. Now this is what a hapten is. This is why it's a little confusing. A hapten is generally a very, very, very small molecule. And remember, antigens tend to be a little bit larger. So this small molecule, if it was to gain access into the body, nothing really happens. But if it pairs up with the protein, a larger protein, in the body, then it causes a reaction. It causes antibodies to be formed. So this by itself, the little triangle, doesn't cause anything. But when I combine it here, then I get antibodies formed. But no reaction. That's why it's referred to as an incomplete antigen. Now, on the second exposure, just the hapten alone will cause a response. It doesn't need this protein anymore for the antibodies to be formed. An example of this is poison ivy. So a lot of times people, they are actually, if you have a reaction to poison ivy, and you say, well, this is the first time I've had that reaction. No, you've actually been exposed to it before. You might not have had the reaction. But it's a very, very small protein, gains access. It has to go through this process to generate antibodies. So on the first exposure, nothing happens. The second time, all you need is just the hapten. And then the hapten causes a response, and that's when you get the itchy. This happens with cosmetics, those types of things. Did you have a question?
I just wanted to make sure that the red represented the protein.
Yep. Guess what?
So on the complete antigen, what did you say the first thing was?
Immunogenicity. It forms antibodies. That's why this one is incomplete, because it by itself did not cause the formation of the antibodies. It doesn't have immunogenicity. It does have reactivity, though. Now let's talk about our two arms, so to speak, of the adaptive system. Humoral immunity, cellular immunity. Humoral are B cells. They are a lymphocyte. They are formed in the red marrow. And there's a term I'm going to use that you need to know. It's called immunocompetent. That is a B cell or a T cell that knows its job. In other words, I told you that there was one good guy for every one bad guy. Well, it has to know what bad guy it's supposed to fight. And not only does it know what bad guy it's supposed to fight, it also needs to know I'm not supposed to kill self. Well, to do that, it has to go to school. Now, the B lymphocytes, they go to school, they go to public school. I'm born in the red marrow, I'm going to go to school in the red marrow. Whereas the T lymphocytes, they're from a wealthier family. They're born in the red marrow, bless you, and they're going to go to boarding school. Boarding school is in the thymus. So they get shipped off to the thymus to become immunocompetent. Remember, immunocompetent is for both of them. It just means I know my job. I know what cells I'm supposed to be looking for. But I have to learn how to do that. Now, we think the way B cells become immunocompetent is similar to T cells, but we know a lot more about T cells. So I'm going to spend the time talking about how T cells become immunocompetent. So it's a two-step process. It's called a positive selection and negative selection. The first one is rewarded because they do something good. The second one, when they do something, if it's what they're not supposed to do, it's negative selection, and they undergo what's referred to as apoptosis or apoptosis, cell death, cell suicide. Now remember we talked about that MHC, the major histocompatibility complex, the flagpole. Okay? So this is a flagpole. This little purple thing here. Okay, so this is my T cell. This is what's referred to as a T cell receptor, and that's an antigenic determinant, or just a piece of the antigen. All right, which would be the antigenic determinant. Now, when you look at the top one, the whole point here is that they have to be able to recognize self from not self. So they have to be able to recognize that major histocompatibility complex. They need to be able to bind to the flagpole. So when you look at this one, does it look like it's binding appropriately? No. So this is going to undergo apoptosis, cell death, because it didn't do its job correctly. This one gets to go on because it does do its job correctly. It's recognizing the flagpole. You see that again, the purple and the green are matching up exactly. But notice how there's still some space by the tic-tac, the antigenic determinant, which is good. It's not recognizing the antigenic determinant. If it binds to the antigenic determinant, that's a reaction. So this is why it's referred to as positive. Now this is one particular type of cell in the thymus. We then bring a different type of cell into the thymus, or from the thymus, and introduce it. So again, if you get to move on to the next round, you didn't die in the first round, I'm then going to do the same process again. So notice we do have binding here. The green and the purple are matching up exactly. But look what happens. Is there any space up here? Nope. So it's actually binding to a self-antigen. It can't do that. So this cell undergoes apoptosis. When it doesn't bind to it, it gets to move on. So this is why it's called a negative selection, because it didn't do something. In the earlier one, it did do something, and that was the positive selection. Here it's the negative selection, and it's not binding, and these move on. That's how these cells become immunocompetent. How they learn their job. And as I stated before, if 100 cells go into this school, only 2 make it out. 98 undergo apoptosis. So 2% of all T cells graduate. 98% die. And that's important because we don't want our immune system attacking our body. So when I talk about pathology, that occurs where we have some individuals that their immune system attacks their body. Okay? It's called an autoimmune disease. Now, how do I know that this cell has become immunocompetent? And this, again, happens up here. So you can just pretty much take what we say here, what I said here. The only difference is these are doing this process in the red marrow. And we don't know the exact mechanism, but we know it's similar to this. The thymus, the only major difference is they go to boarding school. This all happens, or the T cells all happen in the thymus. B cells happen in the red marrow. When they become immunocompetent, that bad guy they're looking for, we have receptors that match that bad guy. So anywhere from 10 to 100,000 receptors on each cell. And its sole purpose is it's got the exact opposite lock and key. So if this is the antigenic determinant, and it's very simplistic view, then this would be what would be on the T cell receptor. So they fit lock and key. All right. Now we do have some other cells that are present here that are part of the system. They're called the antigen presenting cells. And as I've kind of talked about as we go through A&P specifically with things like, what does a prothrombin activator do? It probably activates prothrombin. What do you suppose an antigen presenting cell does? Pardon me? It presents antigens. Some textbooks will call it professional because there's some in the innate system. Just the antigen presenting cells fine. And the biggest ones here are dendritic. Langerhans are kind of similar to dendritic cells, macrophages, and B cells. Those are all examples of antigen presenting cells. What do? They go to the appropriate T cell and say, here, I just ate this. This is what's now inside our body, so you need to mount an attack and kill it. It doesn't kill the antigen presenting cell. Okay? So if I'm just a regular body cell and I'm infected with something and the flag says not self, I get destroyed. When these guys are flying a flag that says this, they don't get destroyed. They're presenting it to the T cell, so the T cells can turn around and say, got it, we're going to mount an attack against this. Why don't they get destroyed? I'm going to talk about it a little bit later, but this is an MHC class 1. It's on all nucleated cells. So obviously it would not be on a red blood cell because red blood cells don't have a nucleus. But antigen-presenting cells have a different flagpole called a MHC class 2, a major histocompatibility complex class 2. Now, let's talk about how these different arms work. We'll hit the humoral component first. Humoral component are our B cells. Now, they basically are going to kind of lay out here in our lymphatic tissues. And things are floating by. And it's not that a B cell goes after the antigen, and they aren't quite sure exactly why this works. The antigen is going to come to the B cell. So the B cells, remember that the reticular tissue has fibers. The fibers were stroma, like that hammock that I talked about. And the cells just kind of lay there. Now, they don't lay there for a long time. They lay there for a little while, and then they leave and they float around the body for a few days, and they land in a different tissue. Okay? It's basically a way for you to increase the chance that an antigen, a bad guy, is going to come into contact. So I'm going to constantly kind of replace the B cells and the T cells that are there. So the red are the antigens, and these are receptors on the B cells. And again, there would be about 10 to 100,000 of these on each cell. Now notice, it's not going to that one. It's not going to that one. Why? That's not their bad guy. So it goes to the middle one, because this is the one that looks out for this particular antigenic determinant, and it binds. So now it becomes active. Once it becomes active, we're going to have clones formed. Now, one clone is going to become plasma cells. And again, these numbers are not, so this is just a representation. It's not like for every one you activate, you automatically get 3, unless it's Christmas, and then we're going to add an extra one because it's a holiday. So this is going to become a plasma cell. Now, I don't get real nitpicky, but to be nitpicky, the cells that produce antibodies are plasma cells. Okay? I'm totally fine, though, with you telling me the B cells produce antibodies. But you have to understand that it really is a cellular division that occurs. So we form the clones divide, and I get plasma cells. And they're going to make antibodies. Now the other thing that I get is, remember we had 3 characteristics of the adaptive system: specific, systemic, and memory. So I get a memory cell. Now at this particular point, somewhat staggering if you ask me, each plasma cell kicks out about 2,000 antibodies per second for about 4 to 5 days. Okay? And then these antibodies just basically float around in the blood. Now, if I get exposed to the exact same antigen, the exact same bad guy again in the future, look at the difference. So here's the antigen, cellular division. Look how many more plasma cells I get. And I also get more memory cells. So when we look at this graphically, here I've got the first exposure. Okay, so antigen, excuse me, antibodies to antigen A, bad guy A, however you want to think about it. Now this is the part that's a little interesting. At T0 is where I was exposed. I'm generating antibodies, but they don't start really picking up for about 3 to 4 days. We're basically going through the process. We just got attacked. Well, who's attacking us? I don't know. I gotta get more information. So the immune system is basically tearing up the antigen, figuring out who the antigen is, and we start manufacturing antibodies. Okay? Now notice, not a lot of antibodies. And they don't last a long time. They start to go down. Now let's say, right at the end of the month, I was exposed to that exact same bad guy. Second time for antigen A. Look at the response. 1, hardly any lag at all. Here we had a big lag between the time we started, time we were infected, and the time we started putting out antibodies. Here, almost none. And look at the response. Huge difference. So obviously, this is the amount of antibodies. Moderate amount, tons. And it lasts for a much, much longer time. But what happens if I'm exposed to antibody or antigen B, bad guy B, at the exact same time? Bless you. It looks exactly like the first one, because it's the first time we've been exposed to that bad guy. So in the first exposure to anything, they will always be identical, the response. Now I will tell you, if we were to look at that last part, so second exposure to antigen A and antigen B at the same time, clinically there's probably going to be a little bit of a difference, because you start to see when we progress through this adaptive system, the adaptive system is going to come back and stimulate the innate system. So in other words, the innate system is going to be a little bit stronger. And so clinically, the way you might present might be a little bit less, because you have a coexisting, or if it's a fairly significant antigen, you might be worse, because you could already be depleted. Does that make sense as far as that response goes? Now, when we talk about humoral immunity, you have different ways of approaching humoral immunity. Okay? The first one is that you earned it. In other words, I got the bug, I got sick, I developed antibodies, therefore, I earned it. That is the active process. You acquired the bug. Now, also under active, I can give you a vaccine. Now, a vaccine is going to give you cellular immunity. So the top one up there where it says active, we're getting cellular immunity here. The first one because I earned it. The second one because I'm going to administer to you usually a dead antigen, or what's referred to as alive attenuated. In other words, I took out the disease-causing part of the antigen, the bad guy, and I give it to you. So your body goes, oh, I'm going to go ahead and I must have been exposed to this, and I'm going to get antibodies as well as I'm going to get cellular immunity. That's how vaccines work. It actually came from a Spanish scientist, and what he did was he noticed that they really did have milkmaids at the time, and he noticed this was a time where smallpox was present. There was a bug that was a pathogen that was similar to smallpox. It didn't cause as many bad things, and it was called cowpox. Okay? And so what he did was he noticed that the milkmaids had pustules or things that emanated from their hands, which is kind of disgusting when you think that they're milking the cow. So he took stuff from there, and he injected this little boy. So he wasn't a very nice scientist. And he then exposed, because what he noticed was the women, the milkmaids, were not contracting smallpox. So he inoculated the little boy, and then exposed the little boy to smallpox, and he didn't get smallpox. So Spanish word for cow is vaca. That's where vaccine comes from. So they were able to then say, I'm going to give you a serum that will cause your body to go ahead and produce these antigens. Not antigens, antibodies, and not just antibodies, but the memory cells. So that now, if you're exposed to it, you're going to get it. It's like the flu. So people say, if I don't want to get the flu vaccine because I don't want to get the flu. You can't get the flu from the flu vaccine. Okay? It's impossible because it's a dead virus. So if you've never heard how it works, basically, places like the NIH, the World Health Organization, they look to see patterns of flus around the world, because they do have this global pattern. So we might see something in Japan before it's occurring over here in the United States. And so they basically look at the 3 most common viruses that cause the flu, and they basically make a vaccine out of that. So do you suppose there's only 3 flu viruses out there? No. They're just taking their odds. So yes, you can still get the flu if you get the flu vaccine, but it's not because you got the vaccine. It's because you developed some other type of flu. They're just, again, taking the odds to say these are the 3 biggest flu viruses that we're seeing, so that's where it comes from. And since we're on the topic, you also cannot get autism from a vaccine. Okay? Most people, and this is the problem with our society where we put someone like Jenny McCarthy on a panel with 3 epidemiologists. Yes, she has an autistic child. Let's put her on a panel to how is it to be a mother of an autistic child. But you don't take somebody who barely graduated from high school and put them on a panel with 3 epidemiologists. But that's what happened in our society. If you don't know, there was a gentleman, there was a surgeon in England, and basically he was studying the relationship to autism and things that happen in the GI system. But what he noticed was that when there were some people that were getting these shots, their GI system was screwed up. Therefore, he made that connection, that it was the vaccines causing it. Well, what they later found out was his study was completely falsified. This man is actually not allowed to practice medicine in England. And then all of the studies that have been done since, not one has ever shown that it causes autism. Back to the point where scientists are saying, stop. We're wasting so much money that we could be spending on other things. So if you don't want to get your kid vaccinated, all I would recommend is that you at least sit down with your pediatrician and you have a heart-to-heart talk with them and see what he or she says before you just go, well, Jenny McCarthy doesn't think I should do it, so if Jenny McCarthy doesn't, then I probably should. So at least if you're not going to do it, make it a fairly well-informed decision. But there is no relationship between vaccines and autism. Now, the second way of getting this is passive. Here, what I'm going to do is I'm going to give you antibodies. I'm not going to let you develop memory cells. I'm just giving you antibodies. So probably the biggest one is mom to baby. Now we can do that through the circulation. So mom is going to impart her antibodies to her baby, and they last for about 6 to 8 months. Then they will eventually get degraded in the baby's body. We can continue that a little bit longer through breastfeeding. Breastfeeding is another way in which you can pass on the antibodies. And again, that's all you're getting here is antibodies. You're not getting memory cells. Now the last way, it's still in the passive process, is through a gamma globulin shot. So if you've ever listened to the news and occasionally you'll hear if you ate at this particular restaurant in the last 3 weeks, you should probably go down to the public health department and we're going to give you a shot because the cook had hepatitis. So we're going to give you antibodies for hepatitis. Again, it's not a vaccine. It's basically just boosting your body's own response before it gets going so that you don't end up with hepatitis. This is what happened with the Ebola virus. So some of the people, some of the medical people that were exposed, we were able to take their blood, get the serum, and in the serum we have just antibodies to the Ebola virus. And what that did was it gave people a running chance at surviving this horrific disease. Again, not a vaccine. A vaccine causes your body to develop its own antibodies as well as memory cells. These are just antibodies, no memory cells. So anytime it's passive, you're going to look at this particular picture here. Active, this has something to do with the bug, with the pathogen. And you will get antibodies and you will get memory cells. Therefore, on subsequent infections, it will look like this instead of this. Here, just antibodies are given, no memory cells. Now we've been talking about antibodies, so let's look and see what antibodies are. Antibodies are these structures that look like a big giant Y. Okay? Now, there are 5 classes of them, and the classes are named based off of this area here. They come from the Greek letters mu, alpha, delta, gamma, and epsilon. You don't have to write them down, but that's what the letters come from. Okay? Now, when we hear the term antibodies, I want you to think of, and I already talked about this in the hematology, immunoglobulins. That's what an antibody is. So when we talk about antibodies, sometimes we refer to them as IgA, IgM, IgE. The Ig stands for immunoglobulin. So if you wanted, you could say it's antibody A, antibody B, or not antibody B, antibody D, antibody M, antibody G. And there are 5 classes of them. Now when you look at its structure, we have an area that's referred to as long chains or heavy chains, and they're the ones that are here on the what's referred to as the stem portion. the leg of the Y. I've got the leg here, and then the arms go out to the side. And then I've got, on the inside, is a continuation of my heavy chain. And then I have two light chains on the outsides of the arms. Okay? This is where we bind our antigenic determinant. So if we go back and look at that one picture, this is an antibody. So here's the stem portion. It's the arm portion that binds to the antigenic receptors. Okay, on both sides. So they're equal. But it is this portion that puts it into its class. So this is another thing that it's difficult for people to wrap their minds around. A lot of people are under the impression that you have to be infected or you have to have an invasion of a pathogen. And that's where we would basically make the recipe to kill that pathogen. Your body is set up to essentially encounter almost any possible pathogen. It's based off of genetics. Okay? And I'm talking about 1 million different antigens. Well, we don't have that many genes in our body. So without getting into the heavy math, a very easy example of this is so that I know that I've got the appropriate antigenic determinants that are going to bind to my antigen receptors on my antibody. If I was to take a deck of cards, and I have to have 3 cards, and those 3 cards equal an antigen, so I take 1, 2, 3, okay, that's antigen A. Come over here, 1, 2, 3, that's antigen B. 1, 2, 3, that's antigen C. You put them all back together, you shuffle the deck, and you do it again. And then you shuffle the deck and you do it again. Okay? Now obviously if you know math, you're still not going to get up to 1 million with 52 cards in a group of 3. So there's some extra recombination that occurs at a different level than this. That increases that ability so that you can make 100 or 1 million different things that will bind. It's just that they don't bind quickly. But your body's already set up for it. It's a genetic process. It's the reason why some people never get sick and other people get sick all the time. Now some of that's you know, your view on life and your psyche and your environment and your health. Other people, it's just their genetics. They just don't get sick. Okay? Now, when we talk about, this is another part that students have a difficult time with. Antibodies have different functions based off of their location, based off of their class. And remember that it's this structure here in the stem region that dictates what their job is going to be. Okay? Now, you can do this whichever way you want. They do spell a word. So MAG, those are the classes of antibodies. So again, we would put IgM, IgA, IgG, IgD, IgG, and IgE. Immunoglobulin A, immunoglobulin M, immunoglobulin D. Now, what does this mean? What are their functions? So this one, IgM, is the first one that the first type of antibody that will be released with the first infection. So when we go back to the slide here, these would all be IgMs. be IgMs for bad guy A. So this letter right here simply designates what its function is. All of these, even though I have 5 of them, can all be looking out for bad guy A. So this one, IgM, will occur at the first exposure. The first time I've been exposed to it, that's what I'm going to see. This is how a physician is able to tell if you've ever been exposed to this bug before or not, because we look at the levels of IgMs. The IgMs are big, and this one here, because I don't want to get ahead of myself, is not big. It means this is the first time you've been exposed to it. Now, IgAs, we find these in the fluids in our body, in our mucous membranes, isn't it?
So the more times they're exposed to the antigen, the number of antibodies goes up or down.
The number of antibodies would go up, but I wouldn't be pointing at that one when you're asking that question. We want to be pointing at this one. Because this is the IgMs of antibody, bad guy A. This is IgGs of bad guy A.
So for the antibodies for the classes, you said they all look out for a certain bad guy, but then they all have like a million combinations to be able to attack different pathogens.
No, the million combinations come from the arm part.
Oh, the stem is what?
The stem is what
Okay.
tells me whether I'm going to be an IgM, IgA, IgD, IgG, or IgE. This is all dictated by the stem region. What I'm binding to, the bad guy I'm binding to, the million, is dictated by the arms. That's the receptor portion. So my IgAs, I'm going to find breast milk, saliva, gastrointestinal tract. It's been found in semen. So it's in the fluid parts of our body. Again, if we're going to use the same example, if I was exposed to bad guy A, I have an IgM for bad guy A. I have an IgA for bad guy A. Can't remember which bad guy. Was it M or A? Was it bad guy A? Okay. So I have if I have bad guy A, I have an IgM for bad guy A. I have an IgA for bad guy A. I have an IgD for bad guy A. These are just classes, and the classes dictate where you're going to find these particular type of antibodies. Now, IgDs, if you go back to this picture, see these things sticking off the B cell? What do those look like to you? Ys, which are antibodies. Those are IgDs, the receptors that are in the B cells, or on the surface of the B cells. So that would be an IgD for bad guy A, and I'm going to find those receptors on B cells. Now, the most plentiful of all of the immunoglobulins that you're going to find floating around are IgGs. And we're going to see these. So they are the second exposure. Notice how there's a lot of them. So when we look at this, if I was to put numbers on it, about 75% of all the antibodies floating around in your blood are going to be of an IgG format. Now, this is the reason that sometimes it gets a little much of a pain sometimes, because I would like to make it simple. I think the simplest way is that IgMs are our first exposure and IgGs are our second exposure. However, this last little bit over here, the very late portion of the first exposure, can be IgGs. So we see them in the second exposure and we see them in the late first exposure. Now, remember I told you that we had, from a passive standpoint, these are IgGs. The ones going from mom to baby are IgGs. So, since I've already presented it, is there another class of which a baby might be able to benefit from mom? IgAs. IgAs are in fluid. They're in the breast milk. So when the baby is breastfeeding, they're getting IgAs being sent to them. And then the last one are IgEs. We see IgEs with allergic reactions. So we have cells, lymphocytes, called the basophil, which we talked about. And I told you that basophils were very similar to another immune cell that's not part of our white cells. Leukocytes, not lymphocyte. And the mast cell was not a leukocyte. It is filled with histamine. So basophils and mast cells work very similar. The receptor on them are IgEs. So when you are allergic to something, and you've already been exposed to it, you will have IgEs stuck in your mast cells and in your basophils that are going to respond to the thing you're allergic to. So we'll talk about hypersensitivities in the pathology portion. Well, I don't want to get in the weeds too much, but that's how you're able to identify it's an allergic reaction. Is I would see an elevated level of IgEs in your blood when you came into the emergency department or the clinic or what have you. And I'd be able to tell that what we're seeing is an allergy.
So then where did the eosinophils come in? Because aren't those allergies?
Eosinophils just happen to be cells that respond to allergic reactions as well.
Okay. So they
These are
don't
specific to the immune system because of the IgEs that are
Okay.
being present there. So when you see this, again, the most abundant, it's the second one. I'd make sure that's in there, and I'd also probably put that it does happen at the end part of the first. The HAs are secretions. B cells, the IgDs are bound to our B cells, and the IgDs are allergic response. All right, why don't we just take a short break, because again, we'll get done a little early. We'll finish up with antibody. The humoral immunity is one to notify the cellular immunity, but also to make antibodies. Antibodies, again, don't kill anything. They aid in other parts of the immune system to do their job. Okay? Now, most of them are going to form based off of what's referred to as the antigen-antibody complex. So in other words, once the antigen has bound to an antibody, that's an antigen-antibody complex. Now, in your book, they actually, I think, do this a fairly good way. And there's only one slight thing that you're going to have to put into your thinking to make sure that this mnemonic works. But basically, plan. If you think of the word plan, so antibodies have a plan for the bad guys, then it stands for precipitation. And what precipitation means is that I'm going to, and it's the one thing I will tell you, and I'm going to show you these pictures here just so that you can get an idea. In your book, you have a table, and the table is going to show you all the classes, so IgM, IgA, IgD, IgG, IgE, and it's going to tell you what their primary role is, which is in blue. Okay, those are the important ones, but I would probably kind of stick with these and just add to the ones I told you, which again, IgG is going to be the second exposure, but it's also going to show up at the very end of the first exposure. They have different shapes to them. So when you look at the IgM, it exists in a a pentamer. All right? Now, why is this important? Well, we have a thing called precipitation. So remember that I can also bind to more than one. So I can have this antigen binding to multiple antibodies. So since it's summertime, if you like iced tea, and somebody gave you unsweetened iced tea, and you're going to add some sugar in there, and you like your tea very, very sweet, more than likely, where are you going to find a lot of that sugar in a little bit? Why? I put too much in. So that's referred to as saturation. But this is essentially what I'm doing. I'm making this so heavy, it can't stay in the blood. It falls to the side of the blood vessel. How is that beneficial? Because then if I am a macrophage, I don't have to go chasing after it. It's not floating around going down the splenic artery. It's sitting on the side. It's basically, I'm puncturing the tires of its getaway vehicle. Therefore, it's much easier than to come by, well, look at this, I'm just going to eat you right here. I don't have to work very hard. So then a macrophage can undergo phagocytosis and destroy these. Okay? And we also remember when we talked about complement, those were those 20 inactive proteins, and we had 3 ways of activating them, all getting to the common pathway of C3. One of the things that activated it, an antigen-antibody complex. Okay, so there's the P. The L, okay, this is where you have to, we're going to add the words in there. So this is lysis. Remember, lysis means to break open and destroy. So part of complement, remember, complement, we pour oil on the situation, opsonins, we increase the inflammatory process, and we cause lysis. So complement leads to lysis. See, that's, to understand the mnemonic, you have to get that part of it. We're going to stick those membrane attack complex in there once we activate C3, and then I'm going to end up with lysis. A stands for agglutination. Remember when I showed you the pictures? We were trying to figure out what blood type people have, and we had the little wells in there. We put drops of blood in, then we added antibody A and antibody B. And if it clumped, if it agglutinated, that meant that if I put in antibody A's, that must mean there were antigen A's that were present there. So A stands for agglutination, and agglutination essentially does the exact same thing that precipitation does. It weighs it down. Since it weighs it down, they can't get away. If they can't get away, it allows for the macrophages, the neutrophils, to come by and destroy it. Yes, ma'am?
What exactly is it weighing down, the antibody?
No, the antigen. Here, we're just happen to be weighing down blood cells because it's one of the reactions we see if I give somebody the wrong type of blood. The antibodies are attacking the antigens on the red blood cells. So if you go back to that slide in hematology, it'll drive that point home a little more. And the last one is called neutralization. And in neutralization, I'm going to block parts of the bacteria or the virus from either releasing nasty stuff from inside, toxins, or I'm binding their binding sites. I neutralize it so it can't do its job. And then eventually, phagocytes will come along and destroy it. So those are the 4 ways that antibodies function. Now, I'll come back and talk about this when we're going to start putting the bigger picture together, because remember, we want to think of these things as being in specific acts of a play. And don't try and figure out why something in Act 6 really has some importance back in Act 2 just yet. Okay? It's a little harder to do. Now, let's talk about our cellular immunity. Okay? So these are our T cells. And we actually have 5 different types of T cells. Okay? The most important ones are cytotoxic T cells. These are the ones that actually do go out and kill infected cells. We have helper T cells. Helper T cells are the ones that run the show. They're the generals of the whole process. And then we have regulatory T cells. These are sometimes referred to as suppressor T cells. They basically keep the immune system in check. Like, okay, you know, we started the battle, let's go ahead and let's ramp the battle down a little bit. Looks like we're winning. And it prevents them from basically getting out of, the immune system from getting out of control. The other two that you don't really have to, well, one of them you already know, which are natural killer cells. But they're part of the innate system. And the last one are cellular T cells. Or, excuse me, memory T cells, sorry. So the three that I really want you to know from this standpoint, because you already know what natural killer cells are because we already talked about them in the innate system, but you should know helper T cells, cytotoxic T cells, and suppressor T cells. Now, because of the receptor, one of the receptors that are on helper T cells, it's called the CD4 protein. These are sometimes referred to as CD4 cells or T4 cells. Because of the protein that's present on cytotoxic T cells, it's called the CD8. They're sometimes referred to as CD8 cells. Sometimes you see them as referred to as T8 cells. The more commonly is CD4, CD8, or it's going to be helper T cells and cytotoxic T cells. Those are the two most common that you're going to see. Now, we talked about how, we talked about these major histocompatibility problems. So when I have a regular cell, a nucleated cell, okay, not an APC, it's not going to be a red blood cell because red blood cells don't have a nucleus. And what happens is we're going to take a protein. Now, when it says that this is an endogenous antigen, it means simply that it's a protein. It's a normal protein inside that cell. And they get broken down all the time. So, a snippet, small peptide, which are just a string of amino acids, are going to get pulled off. I'm going to take them over here. I'm going to put them on an MHC class 1, and I'm going to stick it right here in the cell membrane. So that as they fly by, there's going to be a flag that says, hey, this is us. Do not kill us. Now, if this cell was infected by a virus or an abnormal protein, like a cancer protein, we'd still take a piece of it, stick it on the MHC class 1, put it on the membrane, and it's going to say not self, which is going to be translated that either a natural killer cell or a cytotoxic T cell is going to come by and destroy this cell. It'll be a natural killer cell if what they read is not self. But remember, there's going to be another language right under not self that's specific to that particular antigen, and in that case, it's a cytotoxic T cell that will come along and destroy that cell. Now, if this is an APC, an antigen-presenting cell, so a dendritic cell, macrophage, B cell, then there's going to be ingestion of the antigen. So I'm going to pull it in here. I'm going to make an MHC class 2. So we're going to break that, the bad guy down. I'm going to stick it on the MHC class 2. And then this is what's going to go over to a helper T cell and say, hey, this is what's invaded us. You need to mount an attack. It doesn't say, hey, kill me. It says, hey, you need to go and start making helper T cells and B cells that will kill that pathogen. And again, B cells are not going to kill the pathogen. B cells are going to make antibodies, and the antibodies are going to facilitate precipitation, lysis, agglutination, neutralization. So here's a table from your textbook about just exactly where these major histocompatibility complexes, class 1s or class 2s, are located. So these are going to be on all nucleated cells. And again, our blood cells, not nucleated, therefore they don't have them. Class 2s are going to be on APCs. What is it recognized by? So a T cell, so this is a cytotoxic T cell, is going to recognize this MHC class 1. This is going to be a helper T cell that's going to recognize that. What's going to be present there? Either an endogenous protein, which is normal, that's our self, or virus or cancer, which is not self. Here, it's always going to be something that was not inside the cell, a pathogen. What's it going to say? If it's up there and I belong to self, fine. If not, kill me. This one says, hey, I'm one of you. But what I got on board is something that's invaded the body, so you need to mount the attack and kill it. So that does a pretty good job about kind of encapsulating what the different MHCs do. Now, our helper T cells, they're the generals, they're the ones that are going to start the process. So they have to obviously be presented with an antigen via an antigen presenting cell. Now, this is a little complicated. Try and find the best one. So we know that dendritic cells are APCs. So they have undergone phagocytosis, they're drifted apart. There's the antigenic determinant. It's got all sorts of little receptors on there. So there's my MHC class 1. So here, a couple things have to happen. So my T cell receptor has to bind to my MHC class 1. And how did it know which helper T cell to go? Because there's a helper T cell that matches that antigenic determinant. That bad guy. Okay? Now, there's also a special protein, and it's the way that the cell gets its name, which is a CD4 protein. So the CD4 protein then has to come in and bind. Now, once these two things have occurred, this is essentially turning your ignition to your car on. Okay? Does your car go anywhere once you turn the ignition on? What do you have to do to make the car go somewhere? Put it in gear. So right now, all this cell is doing is sitting in your driveway with the engine running. And if nothing happens, then what's going to happen, well, the next step will be a thing called anergy. So energy except with an A, which essentially is it's a cell that's activated, but it's unresponsive. It will just sit there basically until it's destroyed. So there has to be what's referred to as a co-stimulation, a secret handshake. It's just another way to ensure that we're not activating the system before it's supposed to. Now, it could be chemicals that are released from either one of these, or it could be these molecules. So this particular one we usually see with macrophages instead of dendritic cells. But the macrophage has a B7 molecule. The helper T cell has a CD28. By the way, the CD stands for cluster differentiation, so it's not a big deal. It's just easier to memorize it, but the letters do have some meaning. So this is the secret handshake. So the first thing to put the car in gear, we had to have the MHC class 1 with my Tic Tac. We had to have the T cell receptor that matches the Tic Tac. Then I have to have the CD4 protein that comes over. That turns the car on, but it doesn't activate anything. I have to have co-stimulation. And again, co-stimulation will either be chemicals that are released or molecules that sprout. So that, I like the molecule one because that's the secret handshake. Secret handshakes are a little bit easier to see with molecules than they are with the chemicals that are being released. So it's that process that then puts the car in gear. Now when the car gets put in gear, I make memory T cells, memory helper T cells, and I make more helper T cells. Now, I personally like this particular diagram because it sums up the whole thing. Okay? I told you that helper T cells were what rank? They were the generals. So here I've got my generals, little stars up there. He's got an antigen receptor, my T cell receptor's right there. Here's my MHC class 2 because this is an antigen presenting cell. There's a little Tic Tac, there's my antigen, and again, really that's an antigenic determinant that's present there. So once the two things that you're not seeing here, which is the only negative about it, is the binding of my CD4 protein and co-stimulation. That's the only thing missing from this particular one. Now, when I have those, when I have the co-stimulation, I have cytokines that are being released. Now, this is not the molecules or the chemicals of co-stimulation. They can be. But this is what happens when I turn the car on and put it into gear. Okay? Is I have cytokines, and please don't get lost in the interleukins. Interleukins are cytokines, and there's a bunch of them, but you don't need to know which one is causing which. It's not important. But what you should know is that they're being, the helper T cells being stimulated, and what do I end up with? More helper T cells for that bad guy. What else do I get? More B cells. B cells make what? Antibodies. What's he spitting out?
Antibodies.
Antibodies. And remember I told you, no, let me finish with this part first and then we'll move over to the other side. So here's my cytotoxic T cells, looks pretty badass. I'm going to end up getting more of them. Look, he's threatening the little cell over here. This is an infected cell. That's why it has an MHC class 1, so this is a normal nucleated cell. But I get more cytotoxic T cells for that bad guy. Now, remember I told you that when we had an infection, looking at the secondary infection here, that since I was infected with the same, or two different bad guys, this just happened to be the second exposure. I said that you might not have the same clinical response to bad guy B. It could potentially be less. Why? It's because my innate system, or the adaptive system, goes back and increases the activity of the innate system. So, through other cytokines, I'm going to take my little lackadaisical macrophage, piss him off, now he's ready to go and eat a lot of stuff. And then the same with my natural killer cell. Just sitting here doing nothing, and now he's very angry and he's going to go off and kill. And again, these two are the innate system, which means they're not looking out for just bad guys. So if this was bad guy A up here, and all of these were specific to bad guy A, these are just specific to not self. So when you look at the big picture of the system, that's all these pictures are going to be telling you is essentially binding MHC class 2 binds T cell receptor, CD4, that turns the car on, this little co-stimulation down here with the molecules, the B7 CD28, puts it into gear, and then I make more of this. The more complex ones are when they start adding processes for activating B cells. So these sometimes, when they're referred to as naive, that means they haven't been introduced ever to their antigen, and it's not until they're introduced. So here, this is a kind of the way that the C, or the helper T cell is going to activate that, and then this is just a way that you have them happening at the same time. So don't get lost in this slide. Okay, it's far more confusing than what you need to. You should, understand this picture here. And when you kind of have a grasp of it, that's why this picture is so beneficial, because it shows you everything. Again, the only thing I would add to this particular picture is my CD4 protein, and I would add my co-stimulation. And you can either just write the word co-stimulation, or you can make the little bar of the B7 coming from the antigen-presenting cell to the CD28 coming from the helper T cell. And again, this is showing you the same thing. This is from that previous slide. This is just adding more. But I'm also activating my B cells, I'm activating my cytotoxic C cells, and then I'm going back to Act 1 in my innate system, from Act 5, my adaptive system, and I'm stimulating that process there. Any questions on that? Yes, sir.
So the MHC chains 1 and 2, they're just different. I'm still trying to understand. So they're different flagpoles.
Yep.
For
They're proteins.
Okay.
The only reason I use flagpoles is because I talk about the peptide being a flag that has a, because there's so many different ways of describing it. And one of the ways people describe it is like pirates on an ocean. So if I'm sitting there floating in a boat and I've got a flagpole and I see pirates are coming, I might raise the flag up to say, I'm one of you, leave me alone. So, but it's a protein. The MHC class 2 and class 1 are different types of proteins that are stuck in the membrane of that cell. And again, the only ones that have class 2s are our antigen-presenting cells, dendritic cells, B cells, macrophages.
Okay, I think that was my question.
And then all other cells have class 1s, if they have a nucleus. That's one of the reasons, again, I like this particular table, because it shows you basically where I'm going to find them, what recognizes them, what they have on their flagpole, what the response is. All right, so we'll go ahead and in there today. I'll do pathology tomorrow.